Welcome, listener. You must be interested in the function of the animal cell. My name is Professor Glockenstein. After many long years of study, I am a cell biologist, and I am prepared to teach you all you ever wanted, or didn't want, to know about the animal cell. Also here with me, this is...
Hi everybody, my name is Franz. I'm going to be helping Professor Glockenstein in his class.
Well, let's get going. First of all, an animal cell is a eukaryotic cell, which means it has a membrane-bounded nucleus and separate organelles with specific functions. Each organelle has a job to do, and its job helps the cell to function with the greatest efficiency.
Plants have cells too, but animal cells are more interesting because they have a flagellum and they can move.
Yes, Franz, animal cells have flagellum. Flagellum are long whip-like structures made of microtubules that enable animal cells to move. They project from the cell and wave an S-shaped motion that propels the cell. Microtubules, which make up flagellum, are straight, hollow tubes of proteins that give structure and rigidity to a cell.
So, flagellum are like the propellers on a boat.
Excellent! Now, let's talk about the nucleus. On the board is this structure. The nucleus is the control center of the cell, and it is also the largest organelle. Inside the nucleus, most of the cell's DNA is contained in long fibers called chromatin. The other thing the nucleus contains is the nucleolus. Not to be confused with the nucleus is a ball-like fiber mass that contains the parts that make up organelles called ribosomes.
So, the nucleus is like the steering wheel of the boat.
Yes, I suppose you could say that. The job of the nucleus seems like a fairly insignificant job until one knows about ribosomes. They are the smallest and most numerous organelle in the cell. They are clusters of proteins and nucleic acids assembled in the nucleolus. Their job is to create proteins out of amino acids. Proteins do most of the work in cells, and without ribosomes to create them, the world of the cell would come to a screeching halt. However, the cycle does not end there. After a protein is created, they are inserted through the membrane of the rough endoplasmic reticulum.
Rough endoplasmic reticulum. Are there any other kinds of rough endoplasmic reticulums?
Why, yes, Franz, there are. There are two kinds of endoplasmic reticulum. The rough endoplasmic reticulum got its name from the ribosomes that are bound to the outside of its structure. They work closely with the ribosomes and transport the proteins created in them to different places outside the cell. The smooth endoplasmic reticulum has no ribosomes covering it. A number of different enzymes built into it allow it to perform many functions. One example would be building lipid molecules, which is one of its jobs.
Wow, that's pretty neat.
Yes, it is. Now, after the proteins created in the ribosomes go through the rough ER, they are transported over to the Golgi body. The Golgi body is an organelle that stores, modifies, and routes proteins and other chemical products to their next destination. Once the protein is received by the Golgi body, it is either sent out of the cell to where it is needed, or it is stored for future use.
So, do the organelles just float around and bump into each other like bumper cars at a carnival?
Fantastic question, Franz. I'm so glad you asked. The organelles are suspended in something called cytoplasm. Cytoplasm is a jello-like, translucent fluid that fills the cell. Not only does it keep organelles from bumping into each other, it is also where much of the cell's growth occurs. Pretty neat, huh? This reminds me of another organelle found in the cytoplasm, the centriole. Within the centrosome, it is the area where microtubules are produced. The two centrioles do their job. Centrioles are needed to make motile cilia and flagella, as well as organize the microtubules in the centrosome, when cellular division is about to occur. Also found in the animal cell are the lysosomes. Lysosomes are small, round organelles that contain digestive enzymes. If, perchance, a lysosome was to explode, then the powerful enzymes inside them would devour the entire cell.
That sounds horrible. Are they just useless little bombs in the cell, just waiting to go off and eat up all of the other organelles?
Franz, the lysosomes are anything but useless. Lysosomes combine with food particles and devour them, so the cell can then use their nutrients. Vacuoles, here on the diagram, carry undigested food to the cell. However, the only way the food can get to the cell is through the lysosomes. A vacuole will come into contact with a lysosome, and the enzymes in the lysosome will digest the food particles in the vacuole, therefore feeding the cell. The plasma membrane surrounds the cell and works as a barrier between the cell and its surroundings. It is very thin, in fact, 8,000 of them stacked together would only be as thick as a sheet of paper. The plasma membrane, which is made up of phospholipids, allows the cell to accept only the particles from outside that it chooses. Some particles can pass through, and some cannot.
What if the molecule is really large?
Well, that's where ATP comes in. ATP is the main energy product used by the cell. It allows the cell to move larger molecules through the plasma membrane. Through cellular respiration, it is created in the mitochondria. Unlike all other organelles, mitochondria have both an inner membrane and an outer membrane. The outer membrane covers and contains the organelle. The inner membrane folds over many times and is where many of the chemical reactions happen.
Cells are so teeny.
What was that, Franz?
I said cells are so teeny.
Yes, Franz, but even though they are teeny, they are still incredibly complex.
The cell is like a miniature factory. If one small part goes missing, the whole factory just shuts down.
Yes, Franz, from the nucleus to the tiniest ribosome, everything in the cell is crucial to its function. Thank you so much for watching. This is Professor Gluckentine signing off.